Good afternoon, students. It is right now about 10 till 5. So I wanted to work with you today on Neuro 3. Before we get on Neuro 3, I'm just going to tell you where we've been and remind you of what you know. We started out, we defined the nervous system. We broke it down into a diagram. We mentioned two types of nerve cells, neurons and neuroglia, and defined each. We said there are three types of neurons. They're the ones that send an impulse: sensory, associative, they're also called connective or interneuron, and motor neurons. We discussed receptor organs and effector organs. We talked about afferent structures and efferent structures, and we discussed the anatomy of a neuron. So all of you should know that well from lecture 1. Please keep up with these lectures, because if you don't keep up with the lectures and study them as if you're taking a test the next day, then when we do have the neuro test and the final, you'll be swamped. So I want you to feel real comfortable. So please keep up with these. I did see that there were 33 reviews on the first. So you want to stay with me and study so you understand where we've been and what we're talking about now. The second lecture, we defined neuroglia or nerve glue. And there are 6 types of neuroglia, but we introduced you to 2, the Schwann cells and the oligodendrocytes. We talked about the Schwann cells being in the peripheral nervous system and I defined myelin, which was a lipoprotein and made by the Golgi apparatus, and it's filled full of a fatty lipoprotein that winds its way around an axon. And in the neuroglia, the Schwann cells of the PNS, they also have an outer layer known as the neurolema, which gives them an ability to regenerate. And that's why some nerves injured in the peripheral nervous system can certainly regenerate, not in the central nervous system. The oligodendrocytes do not. You did see a picture of those. They're beautifully illustrated in your textbook, the Schwann cells and the oligodendrocytes. Then we discussed the nerve impulse. We explained the fact that the nerve impulse is a wave of electrical negativity. It's like an electrical impulse because of the inward pull or push for sodium and the outward release for potassium. And they would change their polarities as the impulse would travel down the line to the collateral branches and eventually the neurotransmitters. So that is the sodium-potassium pump, and it requires energy in the form of adenosine triphosphate to keep that pump going. We also discussed, and this was a little bit of a review, the 4 neurotransmitters: acetylcholine, acetylcholinesterase, epinephrine, and norepinephrine. Those are the 4 we discussed. Know their names, know what they do. They are very important. That was Neuro 2. Now today, Neuro 3, I'm going to discuss nerve tracts and nerves and tracts, and define each so that you have a good sound understanding of them. Again, I'll talk about a little bit with the receptor organs and the effector organs. We'll talk about the 3 parts of the brain. I'll mention brain landmarks, and I'll discuss the cortices and the cortex of the brain. So now, if you look in your notes on page 10, lecture 3, it's right there in front of you. Open to page 10, lecture 3. And in your textbook, I would like you to open up to page 421. That's in Hull's new edition of Human Anatomy and Physiology, because I want you to zero in on that cross-section through a nerve. Just like we zeroed in and looked at the cross-section through the bicep brachii, we'll look at the cross-section of this nerve. When we talk about nerves, we're talking about structures that are made up of thousands of axons of neurons. Axons of neurons. And if you remember this picture that we've studied quite a bit, this is the axon of a motor neuron. If you look at a motor neuron with a naked eye, you're not going to be able to see it. You can see it with a microscope, but certainly not by the naked eye. But if you take thousands of axons and you bundle them together, yes, in a fasciculus, we call them fasciculi or fasciculus, just like we talked about bundles of muscle fibers being a fasciculus and the plural of fasciculi. If you bundle lots of these axons together, you're going to get a nerve. A nerve, if it is in the peripheral nervous system, the same structure with bundles of axons in the CNS is called a tract. A nerve in the PNS, a tract in the CNS. Now, the beauty of this is when you bundle thousands of axons together, then you can see them. You can see them with the naked eye in surgery. I did mention to you the other day, and I didn't show this picture. If you look at an axon, and this would be an axon of a motor neuron in the PNS, because you can recognize it because of the Schwann cells wrapped around it. In Neil's diagram, he didn't show a little space between the Schwann cells, but there is a space. And we know that the impulse moves rapidly because in the saltatory jump, the electrical impulse jumps from node to node to node in this little space and does so much faster in a myelinated neuron that looks white than in a neuron that is not myelinated and gray in color. What I did want to show you, and I hope this shows up pretty well. I can see it here, so hopefully y'all can see it. I don't want there to be a glare. But here is the axon of a motor neuron, and it's taken it's a microscopic, it's not a drawing, it's an actual microscopic photo. And right here is the node of Ranvier. And you can see how this Schwann cell tucks in, they're called saltatory jumps in a myelinated neuron. And I wanted you to be able to see that. Now, I want you to also look on page 421 in your textbook. And I'm going to try and hold this up because when you look at 421, they're showing you an actual nerve with thousands of axons bundled together so that you can see that nerve. Now, what's interesting is that Mother Nature doesn't reinvent the wheel all the time. We can use a lot of the same terminology in nerves and a description of a nerve that we used when we described muscles. This entire nerve has a membrane around the nerve, just like your muscles have a membrane, the epimysium. This nerve has a membrane, and that nerve is the epineurum. So instead of an epimysium around the muscle, it's epineurum, neuro, nerve. You pulled out a bundle of nerves, and it's referred to as a fasciculus or fascicle. And when you look in this cross section of a nerve, you see all of the fasciculi of the nerve or the neurons. They're bundled together in fasciculi. Singular, a fasciculus, plural, fasciculi. You bring out one fasciculus, and around that fasciculus, you're going to see a perineum, and it's written right here. That's a perineum. Then you pull out an axon, and around the axon on the outside is an endoneurum. I did say perineurum and endonurum around the individual axon itself. So, and then they point to the nodes of Ranvier. So you see that beautifully illustrated by this artist on page 421. Look at them. You certainly paid for these diagrams, and artists charge quite a bit of money to do these beautifully. And this one is well done. So please look at that. Thank you for doing that. Okay. Now, on your notes, if you follow along with me, individual neurons, as I said, are not visible, but many neurons bundled together form a nerve. A nerve is a cord-like bundle or group of bundles of nerve fibers held together by layers of connective tissue. And just as we had connective tissue in the muscles, we do in the nerves too. The muscles had the epimysium, paramysium, endomysium. The nerves are bundled with connective tissue, epineurum, perineurum, and endonurum. That should be easy now because you've gotten that underway because you knew that about the muscles. Now, those are nerves, but a nerve inside the brain and spinal cord, we don't use the term nerve, we say tract. And that way, when an educator is teaching you about tracts or nerves, you know automatically nerves are in the PNS, tracts are in the CNS, the same kind of structure. A bundle of nerve fibers within the CNS, as you can see, is a tract. And tracts carry impulses to and from the brain. They go up and down from the brain. Now, I do have a photo of that, so you'll just have to bear with me. Okay, here we go. You know, I had to look around for my transparencies when I was teaching y'all. I have to do the same thing on this. All right, let me put that one away, and I want you to see this one. Now, this is a cross section. This is a cross section through the spinal foramen. You know about that intervertebral foramen. Here is the centrum. This would be the front of the body. There's the centrum. Here is the transverse process on one side. They're not showing it on another, but here's the cross section of the spinal cord. Now, when you look at a cross section of the spinal cord, the white area that is around this gray butterfly shaped structure, that is the myelinated portion of a spinal cord, and it's made up of tracts to the brain or come down from the brain and descend. The ones that ascend go in one direction only. They go towards the brain. They're going up towards the brain, and these ascending tracts are bundled together with the epineurum. And those that come down are bundled in the same way. The ones that ascend are carrying messages from the body to the brain. When the brain makes a decision to innervate a muscle, it'll send that message down a tract, a descending tract down the spinal cord, and then out through the nerves to the muscles and glands. Now, there's another picture, and this one is in whole. This diagram is in whole too, so you should be able to find this diagram in the discussion of the spinal cord. Okay. Now I'm going to show you. Here's another nice big view of the spinal cord. Here is the white portion of the spinal cord. This is a transverse cut through the spinal cord. Everything in white, as you can imagine, is myelinated, and the artist has colored these little sections different colors, and each little section has a different name. Some of these are tracts, bundles of tracks ascending to the brain, others are descending. Remember, a track can carry messages up to the brain if it's an ascending track, but it can't carry it down by the same track. A message coming down out through the spinal cord has to come down, it has to descend by a different track. They're divided highway here. Now, receptor organs, this is a little bit of a repetition, but I want you to understand it. We've talked about receptor organs, and we said they are organs that contain sensory neurons, an auditory stimulus for the ear. So these receptor organs have sensory, afferent sensory neurons that only pick up on what they're designated, what they're designed. In the eye, it would be light. In the ear, it would be sound waves. On the tongue, it would be chemicals on the tongue that are picking up the taste of your food. So when we talk about eyes, those organs, those are optic organs, O-P-T-I-C, optic organs. If you're talking about the ears, that is an auditory for sound. That's where they get the name auditorium. Auditory organs are your ears. Your sense of taste on the tongue, and you not only have taste buds on the tongue, but you also have some taste buds on the inside of the cheek through that mucous membrane and under the tongue as well. But the majority of the taste buds are on the surface of the tongue, and that gives you your gustatory, G-U-S-T-A-T-O-R-Y. I always remembered it. She ate her spaghetti with gusto, gustatory sense. If you're talking about the skin, your skin is a receptor organ, and you can feel it. And that ability to receive all those stimuli, that is a tactile sense, T-A-C-T-I-L-E, a tactile sense. Your sense of smell occurs because you have afferent sensory neurons that actually come through the mucous membranes of your nose, and that is an olfactory sense, O-L-F-A-C-T-O-R-Y, olfactory. Your sense of smell is olfaction, O-L-F-A-C-T-I-O-N. And then finally, I wanted to mention one of my favorite senses is the ability of your muscles to send a message to the brain up the spinal cord in those ascending tracks of where your body is in space and time. You can close your eyes and I would say, all right, tell me when your arms are out to your side, when you've abducted them away from the midline of the body. You can do that. You can close your eyes and tell me that because you have a proprioceptive sense that feeds back to the spinal cord and to your brain of where your body is in space and time. That is very, very important. And it's a wonderful sense. And when anyone becomes adept at a sport, ice skating, roller skating, football, what they've done is they've made that proprioceptive sense in those muscles that feed back to the brain. They've done it so many times that there are grooves or neuronal pathways in the brain for the movements that exist in that sport. It's proprioception, P-R-O-P-R-I-O-C-E-P-T-I-V-E, proprioceptive sense, proprioception. And that's mentioned right there in your notes too. Now those are receptors, and that's pretty easy to remember. I do want you to also remember what we talked about when we talked about effectors. Effectors are affected upon by motor neurons. Your muscles, you have over 600 in the body. Each muscle is an effector organ, and all your glands, your salivary glands, your lacrimal glands that give you tears over the anterior portion of your eye, your sedophorus glands, which give you perspiration onto the surface of your skin, and as it evaporates, you carry heat away, you help cool off. These are all glands, digestive glands, intestinal glands. All of these are glands that secrete substances, and they're affected upon by motor neurons to secrete. So effectors are muscles and glands. Now, I do want to mention this to you. Effectors are muscles or effectors that are stimulated by motor neurons to contract, and they can contract on an organ, they can squeeze on an organ, or they can contract on the skeletal system, on the bones, have some sensory neurons in them too. They're primarily effectors, but they do have sensory neurons. If you sprain or strain a muscle, you know it, because there are sensory neurons in that effector that sends the message afferently towards the spinal cord and up the tracks of the spinal cord to the brain, so that you know you're in pain. So your muscle is an effector. Motor neurons are stimulating that effector to contract and move bones, but it is also a receptor in that there are some sensory neurons in there. If there weren't any sensory neurons in these effector muscles, you wouldn't experience pain or discomfort from stressed muscles. And that's very important. That's very important. So actually, those effectors have a double activity there. The contraction of a muscle is occurring because the motor neurons are efferent, coming from that spinal cord. The effectors have sensory neurons in them, which carry the message of pain afferently towards the spinal cord. So the motor nerves are coming efferently to the muscle to contract, and the sensory nerves, which pick up on pain, are going afferently towards the spinal cord to let you have cognition or knowledge of that pain. Okay, those are the effectors. I again break down the nervous system. Now, we've talked about the PNS and the CNS, but I'm going to concentrate a little bit on the CNS. And we said the central nervous system itself is broken down into, we're going to look at the brain with a little bit of focus on structures of this brain. All right, if you look on page 11, I want you to look at the top. The discussion begins as the brain, and I want you to open up your textbook to page 400, figure 11.8. Page 400, 11.8. And I want you to focus on the brain. Let me get some of my brain. Miss having my model, because I always have my model in my hand to discuss it, but you're going to have to use a little bit of insight and just pretend I'm holding it. You can see some wonderful diagrams right there in your text. When we talk about the brain resides within the cranial cavity, and the brain is well protected by that wonderful oval skull. As a matter of fact, the bones in your body are protective of delicate organs, but the organ they're the most protective of is the brain. And you can see that by just studying the skeleton, because that oval cranium totally, completely encases that brain and protects it. So you know there's something vitally important. Brain cells do not regenerate. So if they're damaged, it can be permanent damage. So we evolved with this wonderful cranial case. Inside that cranial case, you're going to find out that the brain is surrounded by three specific membranes. They're membranes, but we refer to them, Greek or Latin terminology, as meninges, meninges, not meningenes, it's meninges. So these three membranes are very protective of that nervous tissue. And in addition to that, the brain is soaked in what we call cerebral spinal fluid. And this fluid surrounds not just the brain, it's around the outside of the spinal cord. The spinal cord is protected too by these meninges, just like the brain is. And the vertebra, the intervertebral foramen, where that spinal cord travels, ascends, and descends, that vertebra is highly protective of that spinal cord, just like the cranium is highly protected of that brain itself as well. Okay. Now, when we study the brain, I'm going to mention a couple of things to you, and I hope you can see this. This is a great diagram, and I would normally, in class, I'd run this off, so you would use this as your model of a mid-sagittal section through the brain, and we would add to it because you're going to learn lots of terms for all these wonderful parts. But we can divide the brain for study, and we always break things down for study. We can divide it into three major parts or four major parts. And anatomists argue back and forth. Remember, if you have two anatomists together, you have 5 arguments. But I'm going to give you both points of view. This is in color, so I like it. You can see this artist is dividing it into 3 parts, the largest being the cerebrum. And it's in green on this. And in metaphysical purple or lavender is what the artist is referring to as the brain stem. And then everything here in turquoise would be the cerebellum. Another word for the cerebellum is the hindbrain. So this artist is breaking it down into the cerebrum, the brain stem, and the cerebellum. Now, other artists and other anatomists say that they would like to break it down. I don't have the other picture, but they like to break it down for study into 4 parts. And the 4th part comes in their perspective about the brain stem. If you look right up here, there is an egg-shaped structure right here known as the thalamus. And on the base of the thalamus is another structure called the hypothalamus. And then from the hypothalamus is a stalk. We'll refer to the thalamus and the hypothalamus, the infundibulum, and the pituitary as the diencephalon. The diencephalon, diencephalon, diencephalon. And then they refer to the midbrain, pons, and medulla as the brainstem. So in this rendition, you see the cerebrum, cerebellum, and the brainstem. And then in the other perspective, they separate the diencephalon from the midbrain, pons, and medulla, and they refer to the midbrain, pons, and medulla as the brain stem. Okay. All right. Now, structures of the brain. The cerebrum, as you can see, is the largest part of the brain, and in Homo sapiens, we have a larger cerebrum than in other primates. So the cerebrum is the largest. Now, the cerebrum is divided into, and this is where I love to have my sample of the brain. The cerebrum is divided into two hemispheres, a right and a left hemisphere. So in this drawing, okay, this would be the left hemisphere right here. If I turn this over, there is the right hemisphere, okay? And these hemispheres, the right and left part of the brain, is divided into a right and left half by a deep, and this is a top view, a deep longitudinal fissure. A fissure is a very deep, deep, deep cleft, and the right and left hemispheres are divided by that. Okay. Now, let me pick up this that was dropped, and you can see that in your textbook on page 401. It shows you the left and right hemispheres, and it shows the longitudinal fissure dividing the right and left hemisphere in half. Now, the outer portion of your cerebrum, if we had a real Homo sapien cerebrum right here, the outer portion of that cerebrum is referred to as the cerebral cortex, and it's about 1/4 of an inch thick. Here's an actual picture of, this would be the right hemisphere, and if you were able to go down just 1/4 of an inch in the cerebrum, that would be what we consider the cerebral cortex. Another word for that cerebral cortex is cortices. That is a cortices, cortices. And it's gray matter. The outside of your brain is gray in color because what you are looking at are millions and millions and millions of cell bodies and dendrites. The cell body and the dendrite has no myelin on it. It's not myelinated because if it were myelinated, it would be more difficult to receive a stimulus. So the outer cortices of the brain is the 1/4 of an inch. It's the gray matter, millions of cell bodies and dendrites. And there's something else to mention too. You have billions of neurons in the brain, billions of neurons, but you have trillions of neuroglia. Remember, you have many, many more neuroglia than you have neurons because they're in service. They protect and take care of those neurons. And there's several neuroglia per neuron. So you're going to have trillions of neuroglia compared to billions of neurons. Okay. Now, if you cut through the longitudinal fissure, if you take the brain and take a scalpel and cut through all the way down through the longitudinal fissure, structure is white in color. Well, that tells you something. These neurons, being white in color, are heavily myelinated. That's why this tissue looks white. This C-shaped structure is known as the corpus callosum, and that is right there on page 11. Each hemisphere of the cortex is connected by a bridge of this white myelinated nerve tissue called the corpus callosum. Corpus means body, callosum means, so it's colossal body. Now, it is a bridge that connects the right and left hemispheres together. It's a cross bridge. And this corpus callosum enables the right brain and the left brain to communicate one with the other. I have read that in females, there are more neurons, myelinated neurons in the corpus callosum than in the male. Would that mean that the female brain is able to talk, left and right brain talk more easily? Well, I don't really know. All I know is that I have heard anatomists talk about the fact that there are more myelinated neurons. And also the neurons, the cell bodies, and the dendrites, they are aimed all the way through this cortices towards the center of the brain, and their axons are myelinated. So that's why you see white tissue in the center of the brain, because these are axons exiting, coming down from the cell bodies and neurons in the cortex towards the center of the brain, giving it that white appearance. That's the corpus callosum. The external cortex, and you can see this in, let me show you. Oh, yeah, I'm showing you this. The external cortex has lots of wrinkles. It looks very wrinkly, and lots of meandering little grooves. These wrinkles in the cortex are referred to as convolutions. 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 And there was a theory at one time in 1800s, the 19th century. There was a theory that the more intelligent an individual was, the more convolutions they had. Well, they've done many studies and on cadavers' brains, and they've studied Homo sapiens who deceased, of course, that had lower IQs or had severe disabilities as compared to geniuses. And no, there were not any more convolutions. So the wrinkles, the number of wrinkles, have nothing to do with intelligence, but those are called convolutions. Now, keeping that in mind, I do want to tell you that Albert Einstein had about 15% more brain mass in the left side of his brain, the left parietal temporal lobes, than the rest of us. So that may have accounted for his genius. He was the genius of the century, the 20th century, and he did have more brain mass. So that makes sense, but that has been noted. He did not want his brain dissected after he died, but the coroner who prepared his body for burial just could not stand to put that magnificent brain of Albert Einstein's into a casket. And yes, girls and guys, he did take it out. He did take the brain out. And he kept it pickled. He kept it in formaldehyde, well pickled. And later on, he let it be known that he did have Einstein's brain. And since then, they've done lots of slices, thousands of slices through Einstein's brain. And those slices are preserved, taken care of, and they've been sent all over the world for study. So he didn't want it done, but I'm glad the coroner did that, because we've learned a lot from studying Einstein's brain. When we say the brain is convoluted. But you also see little hills in these wrinkles. There's little ridges, high ridges that you can see throughout all the convolutions, and then you can see deeper grooves. Well, these have names. The high ridges, one high ridge is referred to as a gyrus, and all of them together, gyrus, Gyrus, is singular, and all of them together are referred to as gyri, G-Y-R-I. The deeper little clefts, they're not as deep as a fissure. A fissure is a very deep, deep cleft. But these little clefts are more shallow. And we refer to each one of these as a sulcus, C-U-L-C-U-S, sulcus, and all of them together as sulci, C-U-L-C-I. So the ridges are gyrus, sulcus are the grooves, all of them together are gyri, and all of the grooves together are a sulci. But a deep, deep fissure, as in the longitudinal fissure, when you pull the brain in half, exposing the corpus callosum, that is a longitudinal fissure, a deep fissure. Don't confuse those. They're important. Now, I want to mention some brain landmarks too. Do you remember when we were studying the skeletal system? We had brain landmarks. You had the acromion and trochanters right back here, the mastoid process, the malleoli on the ankles. These were places on the bones where bone osseous tissue was built up, and where you saw these prominences and processes. Those were areas where muscles were attached. And the muscles were attached by the tendons. The tendons anchored the muscle to the bone. So that's where they were attached. You also have landmarks in the brain, and the brain has been completely landmarked. We will talk just about a few, and there's a few of them in Anatomy and Physiology 1 when you have an overview of the neuro system, as we are doing, where you have to learn some of these landmarks, and they're interesting. Okay, first one, what I'm going to do is, and these are beautifully illustrated again on page 400 of your textbook. Don't let those beautiful illustrations go to waste. Okay. The first one I want to mention is a sulcus right here. There is a sulcus, and this sulcus is dividing the frontal lobe of the brain, the frontal lobe from the parietal lobe, and that is referred to as the central sulcus, and it comes all the way down the left hemisphere of the brain. If we turn this over, there would be, and looked at the right hemisphere, there's the central sulcus. So you have two central sulci on either side of the brain, and they're running at a right angle to the frontal lobe and to the parietal lobe. That is the central sulcus. That's important to know, and I'll tell you why later. Central sulcus. And the next is a sulcus on the side of the brain, and this is referred to as the lateral sulcus. The lateral. It's on the lateral side of the brain, and this sulcus is separating the frontal lobe from the temporal lobe here, the lateral sulcus. And if we looked on, this is the left hemisphere, if we looked on the right hemisphere, we would see another lateral sulcus. I've already illustrated, we've talked about the longitudinal fissure. Now, right here is the fourth. This is the fourth landmark, and that is the transverse fissure. All across the transverse, and it's a fissure because it's deep. And if you remove the outer meninges of the brain, you can actually take your finger and kind of tuck it in from the cerebellum or the hindbrain. Right there that is the transverse fissure. Now, one thing I want to point out to you, and you can see it. You can see it on this diagram. The convolutions on the outside of the hindbrain or the cerebellum are a little different. They're not as convoluted and meandering all around and twisting all around as they are in the cerebrum. They're linear convolutions. And they still have the upper ridge, which is called the gyrus, and then the little valley in between, a shallow groove, which is the sulcus. But these convolutions in that cerebellum are linear rather than convoluted. Another thing I want to point to is the inside, and this is a dissection through a true human brain. The inside of the cerebellum also has white tissue, but it is almost in a tree-like form. It looks kind of like a tree or a bush, and it's a little different than what you're looking at when you look at the inside of the cerebrum, with the corpus callosum and the brainstem all being myelinated. This is different in its myelination. It's still showing you that the outer cerebellar tissue is millions of dendrites and cell bodies, and that the axons are myelinated, moving towards the central portion of the cerebellum. Remember, this is split in half, and you're looking at a mid-sagittal section through the entire brain. Note again, the outer cortex is only about 1/4 of an inch in thickness, and it is referred to as the cortices. Peripheral nervous system, when we talked about nerves, and we discussed nerves and motor neurons and Schwann cells. Now, I focused today on the central nervous system. We introduced the brain, which is the largest portion of the entire central nervous system and the largest organ in nervous tissue. We said that it's beautifully encased in a cranium. It has 3 membranes known as meninges that surround it, and it's entirely bathed in what we call cerebrospinal fluid. Cerebro meaning it's around the cerebrum. Spinal means this fluid is also around the spinal cord. These are 3 meninges, and they hold that fluid in. Your body is constantly making cerebral spinal fluid and constantly removing it so that you have a fresh supply. The amniotic fluid cushions the fetus inside the uterus. That amniotic fluid cushions it and protects it. This cerebral spinal fluid cushions the brain as well and protects it also, along with the meninges and, of course, that cranium. We discussed the cerebrum, which is the largest part of the central nervous system, and we defined the cerebral cortex, or the cortices, which is an outer 1/4 of an inch. And we said you're looking at billions of cell bodies and dendrites in this outer cortex or cortices, and the axons are traveling towards the center of the brain. And that's the reason in a mid-sagittal section, the center of the brain is white or heavily myelinated. And the same thing with the cerebellum. It's white or myelinated. We talked about the 4 different types of landmarks. We mentioned the longitudinal fissure, which separates both hemispheres, the right from the left. It's a deep, deep fissure. And we saw another fissure, which is what is separating the cerebrum from the cerebellum. And that is known, and you can see it right here, is the transverse fissure. We talked about, and you can see the central sulcus right here. And the central sulcus is centrally located, and it separates this lobe from this lobe. And then we referred to central sulcus, lateral sulcus, which separates the cerebrum above, or the, I'll put this up here again because I don't want you to miss that. That lateral sulcus separates the temporal lobe from the frontal and the parietal. And in mentioning lobes, these lobes are colored for understanding, and the color is wonderful because you can point to it. This shows you the frontal lobe, and the artist has colored this kind of in a tan color. That frontal lobe, you have the frontal on the right, the frontal on the left, and it's separated by the central sulcus from this lobe behind it, which is the parietal lobe. Remember the word parietal, it means it's on the side of. Parietal. The artist has illustrated this in kind of a green, and then you have the temporal lobe in baby blue. That's the temporal lobe here. Now, I probably moved my pen a little further down than I meant to, but this one is green. In the baby blue right here is the occipital lobe. So you have frontal, parietal, temporal, occipital. You have 4 lobes in the left cerebrum, 4 lobes in the right cerebrum. Okay. And you can remember this because the frontal lobe is covered by the frontal plate. And remember the frontalis, that is the muscle that inserts on the eyebrow so you can raise your eyebrows. It originates high on the epicranium aponeurosis, but the frontal plate protects that frontal lobe. The parietal plate protects the parietal lobe. Your occipital plate protects the occipital lobe, and the temporal plate protects the temporal lobe. So that makes it a little bit easier to understand those 4 lobes. Okay, that kind of finishes that lecture off, and I did it in 56 minutes. I'm hoping all of you tuned in. I do want to get some idea, because I can see how many people are who's coming up and looking and viewing these. Please listen to them carefully. If you need to, don't hesitate to go through them again. That's the reason I put them on YouTube. You can look at them at 3 o'clock in the morning. And I don't think you can in the webinars, but you can in these. If you have any questions, again, you call me and I'll answer these. We've completed 3 of the neurology. I enjoy being with you much more than this because I just like to see your responses and answer questions right there in person. But if you have any questions, please call me. And until the next time, I will email you now and let you know what we've just done. Call me.